From Microbe TV, this is TWiEVO, This Week in Evolution, episode 38, recorded on December 20th, 2018. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Great to be with you today from Elde Lab Studios.
Last TWiEVO of 2018.
Yeah, it's been a good year of science and podcasting and conversations.
Did about a dozen, right?
Yeah, we're on track.
I'd guess. And next year we will do at least another dozen.
That's right. Maybe throw in a special here or there, depending on if the spirit moves us.
You're going to be visiting sometime next year, right?
Yeah, I'm sure I'll be swinging by.
We're working on a special episode too.
Yeah.
So we always have our listeners in mind.
Looking for opportunities. Speaking of that, Vincent, so I was looking at our picks of the week, which will come later.
Yep.
And I'm just saying, if you're listening to this sort of last minute holiday shopping panic or crisis, we'll have a couple of suggestions for you.
For sure.
To check out on the holiday gift front. So stay tuned or skip ahead if you're in a gift-giving panic mode.
But if you do skip ahead, come back and listen.
Yeah, it's going to be a good episode.
And of course, if you're driving, you shouldn't skip ahead. You should just keep driving.
Let it happen.
Right.
Yeah. That's right.
So today it's me and Nels. We have a paper for you. What do we have, Nels?
Yeah, so I'm excited for this one. So the paper title, will take a little while to explain the details, but the paper title is pretty broad. It's Inhibiting the Evolution of Antibiotic Resistance. And so this comes from Hura Marik's lab. She recently moved from the microbiology department at the University of Washington to the biochemistry department at Vanderbilt University in Nashville. She's part of this new Institute for Infection, Immunology, and Inflammation. And just as, maybe a quick side note, we've mentioned in sort of past TWiEVO episodes, just kind of interesting hotbeds where cool evolutionary scientists and sort of folks taking multidisciplinary approaches to evolution are sort of showing up. I think we're seeing a couple of concentrations. One is in Pittsburgh, University of Pittsburgh. I'd like to, not to pat myself on the back, but I think Utah has some interesting energy. And I would add Vanderbilt University as another sort of maybe surprise institution where there's a growing energy around evolutionary thinking and scientific approaches.
Did you know Hura in Seattle?
Yeah, so we overlapped a bit. She was, as I was finishing up my postdoc and transitioning, she was just starting her lab. Mm-hmm. And she's great. So one of my science heroes, I was looking at the Department of Biochemistry website at Vanderbilt. So they have her listed as new faculty, and there's a sentence in there that I think kind of captures it nicely. It says, she brings a tremendous award-winning science research portfolio, intellect, creativity, drive, and scholarship to Vanderbilt. And I think
whole package.
And I think that that combination of creativity and intellect, that's a powerful one. And Hura has both in spades. So she's a great scientist, great friend and colleague, and a lab to keep an eye on for the paper we'll discuss today, which I think is pretty provocative and interesting, and other projects that they have going as well.
It's a very interesting paper. The evolution of evolution.
That's right. So, yeah, and where does evolution come from? It comes from mutations, and so we're returning. TWiEVO is finally returning. It's been a while since we've really maybe made the mutational process the focus of an episode. Of course, that's happening in all of the work that we consider. But it was TWiEVO 2, actually. I was looking back at that, titled Faster Than a Speeding Virus. We were talking about HIV mutation rates.
Yeah, right. I remember that.
Do you remember? I remember we got in some hot water.
Yep.
Actually, with
John
Coffin, I think.
John Coffin said it was all dicey, I think.
Yeah, and maybe true.
Haven't heard from it since.
Yep. But that kind of, in some ways, that actually might make the point. So, we might think that the mutation rate or just measuring mutations would be one of the simplest things, that would be solved. And yet, this continues to be an active area of research with a lot of surprises, a lot of unknowns. And today we'll be tackling that idea from Hura's paper, but not in viruses, this time in bacteria, and in the context of gaining resistance to antibiotics, which is, of course, a massive both scientific but also topic for the clinic with the emergence of all kinds of antibiotic-resistant strains of bacteria.
So talking about the role of mutation, we'll go back to our previous TWiEVO with David Quammen. A good part of his book, The Tangled Tree, was asking whether that wasn't right, and maybe horizontal gene transfer is what is the fodder for evolution. Right?
Yeah. So it's a great point. For me, the way I kind of think about it is I would classify horizontal gene transfer as a mutational event.
Okay.
Yeah. So right. So
changing the genome, right?
Changing the genome, that's mutation. And so, today we'll mostly be focusing on point mutations, and that's sort of, that seems like the simplest mechanism of mutation. But, I think you can very quickly add into the category. So duplication of a gene, it's a single event.
Yeah.
In this case, you have one gene goes to two. So that's potentially thousands of, in essence, thousands of point mutations at once. Or a horizontal transfer event is sort of borrowing an entire gene or even more in one go. That's a mutational event. But in all of these cases, we're still figuring out those contributions and what it all means.
I think you're right. I think you could classify HGT as a mutational event. And David Quammen was looking at it more in how do you make a phylogenetic tree, right?
Correct.
And that's actually the topic of my pick of the week. So if I don't mention it, remind me.
Definitely. Yeah, we'll circle back on that. And, for phylogenetics, actually, in some ways, the point mutation, and including from the Tangled Tree, Carl Woese, and all of the work with the 16S ribosomal sequencing, that's all, based on point mutations, right? Differences in those RNA sequences.
Yeah.
And the hope is that if those mutations are, neutral or sort of well-behaved, that there's this molecular clock-like function sort of underlying that evolutionary process. And so not only can you see the extent to which different species are diverged from each other, but you can also make estimates about how long that they've been diverged from each other if the molecular clock holds. And that's where things get a little, evolution throws some sand in the gears in the sense that the clock is broken from time to time, or it runs at different rates, and that's, we'll be talking some about that.
That's what I was going to say, Nels. I mean, maybe the clock was faster or slower many years ago, right?
Yeah. And that could be for a lot of reasons. And it will matter as populations diverge. There's a lot of interesting work emerging, revising almost constantly, it feels like, how fast different primate species are, how fast the mutation rate is, or the frequency of mutations are arising. And so, again, a lot going on there. So here, now we're talking about, so we'll be talking about mutations, but again, in the context or framework of antibiotic resistance. So this will be some different strains of bacteria, some familiar faces, some common clinically relevant bugs, and even in the introduction. So the paper leads off, and I think, so we'll have the link to the paper. It's in I think it was published just in the last month or so in Molecular Cell. And the introduction claims at the beginning that we're losing the arms race. And this, again, relating to the fact that we're observing all of these antibiotic resistant strains, that there was a sort of golden era in medicine not too long ago when penicillin was discovered and then applied. And you could maybe, from that view, you might say we were winning the arms race against bacteria for a while. But now the sort of, at least the trend lines look like the reverse is true. All of our antibiotics are starting to fail us, and perhaps faster than we're coming up with new ones. And so there are estimates, I think, where the number of humans that are anticipated to die because of antibiotic-resistant bugs, it's growing rapidly and will soon surpass, on a yearly basis, the number of deaths that are attributed to things like cancer So this is a major, emerging medical issue. Now, from an evolutionary standpoint, I guess we could ask: Are we really? What does it mean to lose an arms race? Is that really true? I think you can also make a counter case, at least from a whole species perspective, that we're doing pretty fine. We're doing okay as a species. There's billions of us, and those estimates are only getting higher. So, in terms of a species, I think we're okay. In terms of individuals in the species, though, that could be another matter. And we're starting to see more and more cases, in the news almost every day. Of the one that comes to mind right now is I don't know if you follow the NFL football season at all, but the quarterback Alex Smith from the Washington Redskins just had a nasty leg break, and then on top of that, was infected, I think, by MRSA, a strain of streptomyces that was threatening both his
Staphylococcus.
Staphylococcus. Yep, missed that one. That is threatening his career and possibly even his leg at this point. So I think he might have turned the corner on that. But this is happening. So in that sense, it is true that antibiotic resistance among clinically relevant bacteria is going to be a bigger and bigger part of our conversation.
Yeah, we are afraid that someday we might not be able to treat anything.
That's right.
Unless we get some alternatives here.
Yeah, and so that I think that sort of motivates this study, and specifically considering, so there are, just as you were talking about a moment ago, Vincent, I mean, there are all of these mechanisms of antibiotic resistance. And one of the most common ones are these horizontal transfers of plasmids that encode antibiotics. A lot of famous cases, MRSA, the Staphylococcus, and other, those are oftentimes coming through the exchange of DNA that encodes those antibiotic resistance functions.
In fact, David Quammen's book, he gives a little history of that and talks about the first recognition of antimicrobial resistance, right? I don't know if you remember that. It was really cool.
I do. Agreed. Yep. Yeah, those really cool mouse experiments at Rockefeller from back in the 30s and 40s, something like that. Yeah. So, and then all the way through Lederberg's, ideas of infective heredity coming online at about the same time. Yep, really, really fascinating. Here, though, the paper is actually considering a different brand or flavor of mutations, which are, the point mutation. So on the bacterial chromosome, and they lift up Mycobacterium tuberculosis, a well-known bug with all kinds of emerging antibiotic-resistant properties. And in that case, at least what's been observed so far has been, as opposed to these plasmid transfers, has been a lot of point mutations that can confer antibiotic resistance to these bacteria. And so that kind of starts to frame the work. And in particular, they narrow down very quickly on a single protein. It's a DNA translocase called MFD, are the initials of it. So this is a protein that it's highly conserved. You find it both in bacteria and in eukaryotes. And it's been described to be involved in nucleotide excision repair. Do you want to help define that one for us, Vincent?
Nucleotide excision repair. So if you have a mutation, a big mutation on DNA, this can cut it out, so that the polymerase could then go back and fix it, basically. Right?
Yeah, exactly right. Yep, that's right. And so this has been probably most of the work is done so the damaging agent is something like ultraviolet radiation, UV damage. And then what can happen, so there's another sort of flavor or specific pathway of nucleotide excision repair called transcription coupled repair. That's what we'll be looking at today. Right. And so what happens is after that damage, the RNA polymerase protein, so this is the protein that comes through as transcription is happening, so the DNA is becoming a messenger RNA. The polymerase is what's actually catalyzing that reaction. As the RNA polymerase is going across the DNA, it stalls and senses the damage in the double-stranded DNA. There's some other enzymes are involved to come in and, as you're saying, to excise the mismatch. And then because you have that other strand of DNA that presumably wasn't damaged, that can be used as a template for repair. So in another, so that we'll be talking a bunch about this MFD protein. It's been studied for a long time, probably 50 years plus, as a lot of the fundamental work in bacterial molecular biology. It's also been described as a quote-unquote anti-backtracking factor for polymerases. And so this actually starts to connect this idea of the DNA repair, and as we'll see, evolution of antibiotic resistance to what's sort of the bread and butter of Hura Marique's lab. And that is this idea of conflicts between polymerases. In particular, Hura has done pioneering work on describing these collisions that can occur between RNA polymerase and DNA polymerase. So as genomes are replicating through the action of DNA polymerase, making copies of the DNA, they're also transcribing their genes. The RNA polymerase is working to make messages. And so what happens if these two, as these sort of enzyme complexes are chugging along the chromosomes, chugging along the DNA, what if they collide? What happens? And she's discovered how this can be a source of genomic instability and may also have some role in evolution and adaptability.
Mm-hmm.
And so it's some really interesting observations about mutation rates at specific parts of the genome where these collisions are more or less likely to happen.
So I guess all this back knowledge about MFD was what led her to suspect it could have a role in mutation rates, right?
Yeah, that would be my hunch too. And so that anti-backtracking role, that's a way of sort of mitigating the chance of these collisions happening between the polymerases. And so that's probably what kind of got them onto this. How they sort of transition in the antibiotic resistance, that would be interesting. I'll definitely have to ask her the next time I run into her. So
it's curious to know how they honed in on this, because it doesn't sound like it came up in any screen. It was just probably just looking at what it did and said, hey, these are pretty simple experiments. Let's do them.
Yeah, exactly. And that's what's one of the nice things about this paper is it's the experiments themselves are relatively straightforward.
Yeah.
But the interpretations, I think, are pretty interesting and provocative. They might be a little controversial for a few people. I actually saw, it was interesting, I saw a little bit of science Twitter dust up just about the title of the paper. So it's kind of a broad title, Inhibiting the Evolution of Antibiotic Resistance. And some of our science Twitter was complaining a little bit that that was sort of an ambitious title. But I think we'll see that that's kind of what the study is driving at. Mm-hmm. Mm-hmm. And maybe we can return to that idea.
Yeah, I think the title's fair, but we'll let our listeners decide.
Yeah, exactly. And weigh in if you want. Maybe drop us a line. It'd be great to hear what people think.
And who knows? Maybe we'll backtrack.
Yeah, that's right. We'll see if it's an anti-backtracking podcast. To be determined. Okay, so as you mentioned, Vincent, the study starts out with some relatively simple experiments. It actually draws back again on the sort of rich history of bacterial molecular biology. So the Luria-Delbruck fluctuation analysis. Do you remember these experiments?
Oh boy, do I? 1943, well, that was 10 years before I was born.
Yeah.
But the fluctuation simply means that if you have a population of bacteria, then you dilute them out and let them divide in, say, other tubes. Depending on where the mutation occurs in the division, in some you're going to see mutations, in other you're not.
Yeah, exactly. And so this was... at the time, back in the 40s, before, DNA was known as the basis of heredity, early days, there was this controversy as to whether, so it was already kind of the notion that eukaryotic systems like ourselves, like that mutations were random. That was kind of starting from a lot of work in fruit flies and related stuff was already pretty well settled. But for bacteria, it wasn't clear. And so there was, this controversy between our mutations induced or directed, meaning that depending on the environment, that's going to influence the mutations, or are they random the same way that, and so that's exactly, so that fluctuation assay allowed Luria and Delbruck to sort of infer that it was actually random, so that if you, as you're saying, as you dilute these things out, you get a distribution of, in this case, I think it was resistance to phage, T1 phage, if I'm remembering right. Yeah. And so the alternate idea that it was directed, you would have predicted that every time they diluted it out, they would get roughly the same number of resistant bacteria arising, but instead it was all over the map. Sometimes you'd dilute it out, you'd see many resistant clones to the phage, other times there'd be none, and then everything in between. And so that was sort of the first strong evidence that bacteria also are sort of governed by random mutation, with, of course, with selection acting. So kind of returning to that view, so the first set of experiments here involve several different bacterial strains or species. I think Salmonella comes into the mix. There's the Mycobacterium tuberculosis. We'll see Bacillus subtilis and one or two others. And it's a really simple experiment. So that MFD protein, the translocase, is either present or absent from the strain. So you either have a wild type or a knockout strain. And then in this case, the wrinkle on the Luria-Delbruck analysis is instead of resistance to T1 phage, it's resistance to several different antibiotics. So rifampicin, and there's a few others that I think we'll mention as we continue. And so the sort of consistent observation among these diverse strains was that in the MFD mutants, the ones that lacked the gene encoding the MFD translocase, you'd see 2 to 5-fold lower emergence of rifampicin resistance. And in fact, so the molecular basis of this is known. And so you can go and look at the mutations and measure mutation rates. So how many times do you see these resistance mutations arising per number of cells per generation? And that's what's measured in the first figure, or displayed in the first figure. And then they also, you could say, okay, well, that's cell culture. What about, so a context closer to a real infection where there's more variables? And so they went ahead and did some cell infection modeling experiments using CACO2 cells. So these are a colorectal cancer cell line that you can infect. In this case, I think that was a Salmonella strain. And then again, this was Salmonella wild type or MFD mutant. And there, if anything, the result was even more striking. So the mutation frequency in the knockouts is much lower than in the wild type. So without MFD, the ability of these strains to become antibiotic resistant is compromised, sort of across several different strains and also across several different, or a couple of different experimental schemes.
Now, if you didn't know anything about MTB, at this point, you could argue that it may be a pump that simply is pumping out antibiotics and making the cells resistant, right?
Yeah, exactly. Or, something like maybe the cells are just sick, like it's a general...
Yeah, sure.
You're a mutant and mutants are a little bit sick or something like that. So yeah, so that's sort of, and that will come up in some of the later experiments. And they are, I'd say they are definitely drawing a lot on what's already known about the translocase activity, the role in the nucleotide excision repair. But interestingly, the result that they saw is the opposite of what was seen in some previous experiments, some old experiments. So here, they're actually seeing that you're getting fewer mutations. MFD, I'm looking for where I wrote this in the show notes here, actually is named. So the name of MFD, did you come across that?
MFD.
Yeah. I wrote this down. Maybe I accidentally deleted it.
No, I didn't actually.
Yeah.
It'll come.
Here it is. Sorry.
Go ahead.
MFD. So it stands for mutation frequency decline. So that, so all these, this is the alphabet soup, right? It's named after the phenotype in a lot of these cases.
So that had been known already, right?
That was known. But so the reason it was named that is that if you took it away, it actually, you were more mutagenic, you were worse at the excision repair after an exposure like UV radiation. So
you're more mutagenic. It's the opposite of what we're seeing here, right?
Exactly. Yep. So, and that's why it stands for mutation frequency decline. So it's named after the function. Yeah. If you have it in place, you're actually less prone to with that other kind of damage, UV exposure. So this is the reverse. You take away MFD, and you actually see a mutation reduction.
Yeah.
If you have it in the presence of UV, you see a mutation reduction. But if you take it away, then your repair is worse, basically, right? And so the key difference here, and this is kind of an important point of the kind of central to the paper, is that the kind of insult that you're suffering is going to have a different outcome for this protein.
Right.
And so this is, and that's, I think that's what this paper kind of hinges on, is this idea that, absolutely, if you think about the insult being UV radiation or other things that can damage DNA, then you need this thing to keep things going along to repair the problem. Whereas if you think about the insult being an antibiotic, now the idea put forward is that this thing actually promotes mutation so that you can adapt, or that you're more likely to hit on random mutations that will allow these things to emerge. Yep. Okay. Yeah, I just missed that in my notes. So, okay. So moving forward then, this was, those fluctuation assays or the variation on them. That's, you do that in a really quick amount of time. So you have these massive populations and test tubes of bacteria, and you can move on these experiments really quickly. So what they wanted to do next was allow the experiment to kind of go over, I think it's maybe, 50 generations plus or minus, do some experimental evolution. So expose, again, these sort of the set of bacterial strains, diverse bacterial strains, that are either wild type or lack MFD, expose them to a couple of different antibiotics and different amounts of antibiotics over this longer time, and see what happens in terms of the emergence of antibiotic resistance or mutations that confer antibiotic resistance. And so for two of the strains, Salmonellus and Bacillus subtilis, I think they report that resistance levels were 6 to 20-fold higher in the wild type compared to the MFD mutants. But what really caught their eye was the TB, so the Mycobacterium tuberculosis, that in the wild types gained up to 1,000 times more resistant in the wild type strains compared to the mutant. And so this might echo on for TB in particular, where a lot of the resistance is through chromosomal mutations. That is, you're kind of finding the sweet spot for where having MFD, if you're a bacteria that wants to resist rifampicin, that's an important tool to have as a mycobacterium cell. Okay, so then kind of getting back to your earlier question, or your really good point, Vincent, about, well, is it just a pump that could, just, if you take that away and you can't pump out the antibiotics, that kind of stuff. They start to zero in a little bit on the mechanism. We'll see at the end, they concede that there's more work to be done there. But they start by actually first asking sort of a related question, which is, can they complement that activity of MFD from different strains? So those bacteria are like really highly, different from each other. The MFD is a conserved protein, conserved gene. So can you swap that between different strains? And so they decided to take the Mycobacterium tuberculosis version and see whether that would rescue the ability of the Salmonella that lacked their own MFD to then to have that sort of frequency of antibiotic resistance to rifampicin. And the answer was yes. So you could sub in basically the gene from a distantly related strain. They also now it kind of starts to get into what is MFD doing. So part of its mechanism as a translocase, it's kind of hanging out right on the DNA strands, and it in fact has an interaction with another alphabet soup gene, RPOB. And this, I for some reason I pronounce it ROPB. Is that right? Or am I just? I mean, it's not how it's spelled. It would be, but what?
I've heard it ROP. No. It's just a Ripo or something like that.
Yeah. Anyways, this other gene. It's an RNA polymerase subunit.
Yeah.
And here's where things, they don't make a point of this, but it is a little bit tricky here. So the molecular basis of resistance to rifampicin are just point mutation, or a lot of the known resistance that's been observed again and again are actually point mutations in RNA in this exact RPOB subunit. And so the fact that MFD is also interacting with it does raise the question, is this like, really a global effect on just random mutations that could then, from some set be selected? Or is there, a kind of a more specific interaction already between RPOB and MFD that makes it specific for rifampicin?
Mm-hmm. Mm-hmm.
They don't really express that, but they do, or they don't really address that, but they do look at another antibiotic or a couple different antibiotics, and they see similar trends. So it looks like it's perhaps a more universal mechanism, but maybe a little more ground to cover there. And so in order to actually get at that idea of how often are new mutations arising in the presence or absence of MFD and in the presence of antibiotics, they do some, instead of just looking at the mutations that are arising in RPOB specifically, or there's another gene, Fol A, which is also where there are known mutations. They do whole genome sequencing. So they take 6 replicates from their experimental evolution, the courses, the 50 or so generations of exposure of the wild type bacteria or the knockout MFD bacteria to rifampicin and I think trimethoprim is the second one. And then using that whole genome sequencing, they can just ask, they can compare between the wild type and knockouts. Are there different sort of complements of mutations that arise? And so what they see is the wild types have a much higher abundance of mutations again in the RPOB, in this other gene Fol A. But then they start to find some other targets or some other mutations that appear to be selected on. And one in particular caught their eye. That's in the gene called DNAQ. If you run into this one, I'm talking to a virologist here. Although you did just record an episode of This Week in Microbiology, so maybe you're in a microbiological mood on the bacteria.
I've heard DNAQ, yeah.
Yeah, so that's a pretty well-known bacterial hypermutator allele.
Mm-hmm.
So basically the idea is you make if there are certain mutations that have shown up in DNAQ that then cause the hypermutation. So now the genome as a whole becomes much more insulted with mutations in shorter amounts of time because of that change in that function. And so this is what they observe actually. So some of those wild type replicates become hypermutators by virtue of mutations in the DNAQ, where they never see that in the knockout of MFD. So taken together now, you end up with sets of mutations that are more frequent and sort of they appear more dispersed in the wild type strains than in the knockout. And this is now of the MFD, and this is now under for diverse different bacterial strains, and it's also for several different antibiotics. And so the implication is that without MFD, on exposure to various antibiotics, that the sort of amount of mutation is lower. So that MFD is allowing, in these exposures, it's allowing more mutations to emerge, or it's facilitating mutations. The bacteria have this gene perhaps so that they've evolved this version of the gene or this function so that they can evolve better. They're evolving to evolve is the idea.
So in the presence of MFD, do we see mutations everywhere or just in certain areas?
Yeah, I think, and this is where I had the same question. I haven't looked deeply, but I think this would be an area that I would suggest it would be that would be worth probing a little bit more deeply. Is to really, so they don't mention that, like that there's just more mutations kind of all over the place, and part of that my suspicion is that part of that is that so in these populations, any given point mutation that sort of doesn't confer antibiotic resistance, that's going to be pretty rare. And it doesn't, so the difference between the ones that are selected on, because they confer some of this advantage, they very quickly sweep up in proportion, in frequency in the population. So those are sort of the easiest to observe.
But you would argue do deep sequencing.
Correct.
The wild type and the mutant, and compare them, maybe in the absence of selection, right?
Exactly. So that's exactly right. So those kind of experiments, which are known as mutation accumulation experiments, where you try to put as little selective pressure as possible. I think that would be a really fun experiment to see the results for. Would be whether you take the wild type or the MFD knockout, don't put any selection on them, let them run for 50 plus generations, come back, do deep sequencing, and see whether there are noticeable differences in the random mutations that are accruing just sort of as a normal course of bacterial division. Yep.
I mean, that would be a straightforward result in that this MFD is simply increasing global mutation, and then that makes it easier to get antibiotic resistance.
Correct. Yep.
Now, so what's the role of DNA-Q? Now, DNA-Q is a hypermutator, right? So if you hit DNA-Q, then you get a higher frequency of resistance to antibiotic, right?
Yeah, exactly. And so, but you're also you're kind of scrambling the whole deck, right? So, in DNA-Q, just as for background, it's a subunit of DNA polymerase. And so as the genomes are replicating, this is, there's all this proofreading that's going on so that so that mutations the mutation rate is kept at a really low level. What happens is in some of these mutations in DNA-Q that some of that proofreading goes away. And so it's a much more sloppy process of replication. And so then that's sort of a complicating factor, right? So it's almost this idea, if MFD is increasing mutations, then you're more likely to hit on a mutation that will increase mutations even more. It becomes sort of this two layers or something, or more indirect. Yeah. And so, I think that's a fascinating, and a lot of people have thought about that. Again, this idea of sort of adapting to be adaptable.
Yeah.
Sure. And the trade-offs that can come with that, right? So there's all kinds of downsides to being, so especially in virology, right? So the idea of a error catastrophe threshold, some of the great work from Carla Kierkegaard and Julie Pfeiffer and others, in poliovirus, for example, there's a threshold where if you go over it and you have too much mutation, then the virus populations crash.
Yeah.
You might kind of use a similar viewpoint for thinking about hypermutators like DNA-Q. If this is making it more likely in the short term that antibiotic resistance emerges, those things might be around and under this strong stressor. But for business as usual as a bacteria, these generally they're not hypermutators that are persisting for a long time because of the trade-off or the drag of the mutational load that comes with that phenotype.
So in general, the RNA viruses that have been looked at, they seem to exist very close to the error threshold. That is the number of mutations per genome above which you go and you're dead. You're not infectious, right?
Yeah.
The DNA viruses seem to exist orders of magnitude below it. You have to add many more mutations per genome to disrupt viability in those. But what is interesting is in the RNA virus, at least in poliovirus, that conclusion was reached using a chemical that's a mutagen, ribavirin. And you add ribavirin to virus, you need 1 or 2 mutations per genome to knock down infectivity way low because you're already over the error threshold. If you select for viruses resistant to ribavirin, the mutation is in the RNA polymerase.
Yep.
And it's not in the catalytic site. It's a different part of the enzyme that seems to regulate fidelity. So you could imagine that the polymerases themselves could regulate evolvability, right?
Yeah.
Or in this case, it is something that associates with polymerases, right?
Exactly. Yep. That's what MFD is doing.
And that makes perfect sense, I think.
I agree.
It doesn't have to be the polymerase, right?
Yeah, that's right. Yep. And so, and that actually is the next part of the study is so to get at, and I think this gets a little bit at the specificity of this function. And so because it's well known that MFD interacts with RNA polymerase, the B subunit, they test a single point mutation in MFD. So this is a loose, it's called L499R, meaning that at amino acid position 499 in the polypeptide chain, that leucine, that is the wild type residue, is point mutation causes it to be an arginine. So that's the shorthand there. And that mutation actually breaks the interaction between MFD and RNA polymerase. And so what they can do then is test whether that mutation can rescue the knockout for the property of antibiotic resistance emerging rapidly, or that these mutations are arising. And so they do that. They just simply build, I can't remember which bacteria they used or if they used multiple, but they put back in the L499R mutation and actually see that they don't complement the mutagenesis phenotype of the knockout. Mm-hmm. And so what you can conclude from that is that interaction with RNase polymerase matters for MFD's ability to sort of support the process of mutation here, the emergence of resistance.
What do you think it does? It bends the polymerase a little, so it doesn't is not quite accurate.
Yeah, good question.
Or something else, right?
Yeah,
we don't know.
Yeah, we don't know. I was kind of thinking about it, and we'll talk at the end, but, like, is this process itself regulated? So is this like a clutch? Yeah. If we think of the sort of the RNA polymerase as an engine of, mutational generation, in a sense, is this a clutch, a way of like modulating that somehow? So that, way, who knows? But anyway, so they do the last experiment, and that's actually, it's a relatively short study, but the last experiment is to do a similar thing. But so there's one other known interactor with MFD that's been described, and it's a protein called UVRA. So it's well known for a role. It's an endonuclease, so it cuts DNA. It's known to be involved in DNA repair. So the name, I think the UV R is ultraviolet repair. And MFD is known to interact with that as well. That's at a different surface. So now the mutation is an R165 to A. That's an arginine to an alanine mutation that breaks the interaction between MFD and UVRA involved in DNA repair. And basically they do the same complementation assay or test from the knockout, and it also doesn't rescue. So MFD needs to interact with RNA polymerase, also needs to, or it needs to interact with UVRA, this endonuclease, in order to confer the emergence of these mutations that give antibiotic resistance. So that gets, it doesn't go all the way towards a mechanism, and they concede that in the discussion, but it does, I think it starts to narrow in on what's sort of a combination of what's already known about how it works, and that there is some specificity here. So it's not, just like a drug pump or something else. It really is acting. It looks like it's kind of in the mix with these complexes of proteins that are involved in DNA repair, are involved in transcription. So that kind of gives it some specificity.
They don't really talk in the discussion about what they think is going on. They talk about antibiotic resistance and some other things, but I'm interested in their thoughts for what the mechanism is of MFD's activity, its interaction with these two proteins, and how is that mediating mutagenesis, right?
Agree. So they do launch a couple ideas I thought were kind of interesting. So first of all, just to kind of close on the major conclusion. So they make the case then that MFD is what they're calling an evolvability factor, meaning that it's functioning to increase mutation rates in the presence of these stressors, in this case, the antibiotics. So you're right. So they say that the mechanism, or they don't know the mechanism. It's still foggy, but they had a couple ideas. So one was that it might repair a site that's not damaged.
Yeah.
So basically, exactly. So this nucleotide excision repair, the canonical idea is that you fix something when it's broken. But what if you fixed something when it was already fixed? And so an MFD basically is mediating that action that for some reason now at rate, even though there's no damage at all, you sort of fool the rest of the complex into fixing something that's not broken. And by doing that, you sample more mutations, and you're perhaps more likely to hit on one that will confer antibiotic resistance. The other idea that they float is whether or not could this be inhibiting the action of the related repair pathways. So, and I think that sort of emerges from the UVRA interaction, which is already known to be that interaction, something known to be involved in DNA repair. Does this somehow interact in a way, because you depend on that interaction, does that then sort of turbocharge DNA repair? And again, you're repairing something that's not broken. You're increasing the activity. So I think both of those ideas are at least certainly consistent with the evidence so far. But that would take, a whole nother study in order to narrow in on whether those hypotheses, hold water or false, or to falsify those hypotheses, basically.
And even then, it doesn't say exactly how MFD is doing this, right?
Correct.
It says what processes it might be involved in, but the actual mechanism would be really interesting to sort out.
That's right. And so I think there, you'd really want to link the specific mutation that arises to like sort of before and after, so like, is it, can you sort of show, and you can, this might be very doable. So especially when you have, because you're doing experimental evolution, one of the awesome things about that approach is for bacteria, you can save the whole experiment. Like you can freeze down your parents and grandparents and great grandparents of the bacteria and then thaw them out later. So what you could potentially do is, so if you know that sort of the end of the story is that there is a key mutation in one nucleotide that conferred that resistance, if you went back in every, sort of ancestral generation, you could trace to the exact, really close to the exact generation when that mutation arose. And then, ask whether it was, if it was, fixing something that wasn't broken, or if it was, if it's fluctuating between different mutations at that site, there are some possibilities for sort of tracing, and connecting the mutational process. That could be pretty cool. The other related idea that they bring up, which I thought was interesting, got me thinking a little bit, was a problem that bacteria have so if antibiotics are working against them, what's happening is they can't replicate as well, right? So you're jamming up their RNA polymerase so so that the bacteria can't make more messages, or for a lot of antibiotics, you're really slowing down their ability to replicate. And so what they point out is that because MFD is actually working with the RNA polymerase subunit or interacting with it, transcription in a lot of these cases can actually proceed. Even though the population can't divide a lot, it can still make messages. And so they point out that this transcription coupled repair, by tapping into that it might be a route to sort of continue to generate mutations in the absence of replication. So most of the time, how do point mutations arise? It's you replicate your entire genome and you just make mistakes doing it. And so because you're not replicating a lot, you're not making as many mistakes, but if you're still transcribing and you're somehow modulating or altering transcription coupled repair, and maybe that's what MFD is doing here. It sort of gives the bacteria another escape route, a different mutational pathway. Yeah.
Now people may be wondering why bacteria would have something like MFD that increases mutation. So let's do a thought experiment. If you took these bacteria lacking MFD and put them in competition experiments with wild type strains, who do you think would win?
Yeah. That's a great one. I would guess that MFD would win. And part of the reason it's conserved. So all bacteria have it, or not all, but most.
Yeah.
But it's non-essential, which they point out. That'll be sort of an important point in sort of their conclusions. But yeah, so that you could do those experiments, and actually even as a thought experiment, it's interesting. But you could execute that. Easily,
you could
right?
do it.
You already yeah.
So I would have the opposite thought. I think they would lose because they can't sustain the mutations they need to, exploit a competition or whatever environmental conditions might be present. And the reason I say that, which could be wrong because it's with an RNA virus, if you take the poliovirus is resistant to ribavirin, okay? So they are not pushed over the error threshold by the drug. Those polymerases with that mutation actually are less error prone because at least for polio, the main protein that we know of that gives rise to mutation is the polymerase, right?
Yeah, exactly.
I mean, there may be other proteins involved, but nobody's found them. So you have now a polymerase with greater fidelity. If you put that virus in competition with wild-type viruses, they lose every time, whether it's in cells or in animals.
That's right. Yep.
And so even though MFD is non-essential, I would predict in any competition scenario it would lose because it can't adapt fast enough.
Right. Sorry, that's what I meant to say.
No, okay, sorry.
So yeah, if you lack MFD,
you would lose.
Yeah, sorry. Yeah, that's right. Yeah. Yep.
Otherwise, it wouldn't be there anymore, probably.
Yeah, I mean, I think it's a great experiment. It's a great idea.
Pretty easy to do.
Yeah.
Nels, do you think MFD then would be a good drug target?
Yeah, so that's exactly kind of how they conclude. They propose that and being a little bit provocative, this idea of anti-evolution. So if MFD has evolved to promote evolution, then the notion is instead of targeting all of the, all of the essential, like the RNA polymerase subunit itself, where you see the rifampicin where it's acting, and then mutations rapidly arise. I mean, that's massively strong selective pressure, right? You're dead if you don't get around this. And so those mutations arise quickly and repeatedly. So the idea here is that, as you're pointing out, so that MFD is non-essential, and that perhaps this could be a complementary way to target antibiotic resistance instead of a straight-on antibiotic. Basically, if you could do the equivalent, if you had a small molecule or a drug that would be the equivalent of removing MFD from the equation, then you would basically get the same output that they saw from their experiments, that the emergence of antibiotic resistance and the level of resistance. So remember that 1,000 times difference from their Mycobacterium tuberculosis in the mutant, that you might enjoy some of that protection. And they say that this would be an anti-evolution strategy. You're basically blocking the ability of bacteria to evolve.
And I think they suggest you use it together with an antimicrobial.
Correct. So I would say that second part where you use it together, that to me feels the most robust for that idea, that you kind of hit two different things, or kind of hit it orthogonally at the same time, that you might gain some advantage there. Doing it alone, I'm a little skeptical. So in this idea of anti-evolution, I guess the way that I have been thinking about it is more that, well, evolution happens, and you might come up with strategies that are sort of evolution resistant, that you might buy more time. But the question becomes, so if you target MFD with a small molecule, could then a mutation arise that is resistant to that molecule in the first place, and you're sort of back where you started? Now it could be evolutionary resistant in the sense that because you're targeting MFD, which it's promoting mutations, it's going to take longer for that resistance to emerge. But in terms of sort of the protein interaction, or antibiotic bacteria interaction, that's still, I think that logic still holds. So I'm a little bit skeptical of the idea of anti-evolution, but I am totally on board with the idea of evolutionary resistance, and then mixing these things together, or buying time, or hitting it sort of two at one or three at one. I mean, that's the, sort of the logic behind triple cocktail therapy and HIV treatment. The sort of standard of care today is that if you have any one of those three pharmaceutical interventions to block HIV, the virus will very quickly adapt around it. But if you put all three at once, the probability of a virus becoming resistant to all three at once is sort of vanishingly small, and that's a decent treatment.
I wonder if MFD regulates mutations in its own coding sequence.
Yeah, I love it. Sort of like the snake eating its tail or something.
In that case, so if you treat with a drug that blocks MFD function, small molecule, and then you would decrease the likelihood that you would get mutations in MFD, which would be resistant to the drug.
Yeah, no, exactly. And
right?
I think that's the idea of this anti-evolution. It's like that you target the very thing that you need to generate the diversity.
But the concern is that you'd get mutations in MFD that block drug binding, say. But if MFD mutation is regulated by itself, you wouldn't get those. But I don't know if it is, because as we said earlier, they haven't done the whole genome sequencing.
Correct.
To look at that.
I mean, if you were really clever as a bacteria, strategy, you would actually use MFD as a trap. So if MFD was targeted, it'd be like a tripwire, so that would cause some other mutational, mutation promoter to come online somehow. And it would be sort of the next leg of the arms race, in a sense. where if you target that, it would specifically like cause another process of mutation to like a hypermutator phenotype to emerge. Secondarily, you could like natural selection could, now you have like these multiple levels basically of natural selection. If that was something that had happened, in evolutionary time, or if that's an intervention that, comes to, the clinic over the long haul, something like that could emerge as well. It's always kind of fun to think out the arms race many steps ahead. Because go ahead.
This idea of regulating mutation frequencies, it's an idea for RNA viruses because of ribavirin, which for multiple viruses, not just polio, does push the polymerase over the error threshold. So instead of inhibiting, a function, you just mutagenize the virus out of existence. And, no one has got a drug like that in people, but people are thinking about it for HIV, for example, and other viruses. So it's not the same as this. Here we're reducing the error frequency, but it's getting away from the actual enzymatic function or whatever it is that you're inhibiting and just saying, well, we're going to make a lot of mutations and inactivate the virus. Now, in this case, it probably wouldn't work because these bacteria are existing way below their error threshold.
I think that's right. But until you hit that hypermutator. So, you could actually imagine maybe treatments going or targeting MFD in both directions. So if you could turn it off so that it was gone, or if you could
crank it up, right?
Exactly. So that's right. And then that would be closer to that sort of trying to push the virus population over the threshold. That would be the same for the bacteria. It's interesting to think about it in both directions. I think that all that work in poliovirus is really, kind of raised that idea.
It did.
It's really cool.
It introduced this whole idea of error threshold, right? And it provided experimental evidence, and that can be extended to other systems as well. So that's the value of these model systems, right?
Absolutely. So then, the other question is that we touched on a little bit, is MFD itself regulated? So is this something that is under sort of transcriptional control itself? So for example, if there's a cost, if there is a threshold or a cost to having MFD around, maybe it's turned off when there's not an antibiotic around or another like massive stressor. And then if it was transcriptionally regulated by a stress response, then it would be sort of like turning this thing on right when you might need it, right when you need sort of like made to order mutations or something like that.
Right.
So that would be I think that would be another interesting direction to think about or pursue. Maybe they are already have looked at that, or that's the next chapter, or maybe that's just sort of a fanciful idea. So, it could be getting back to your competition experiments. The other thing, maybe I'll hedge my bets slightly and say it could matter if you're a wild type or mutant, depending on the environment you're in.
Yes.
So, right. And so maybe if like things are like things are good, there's no antibiotics around, there's tons of resources around, then maybe being the mutant is better. That you're right. Who knows?
I could see that. Yeah, for sure.
Yeah. In competition, that would be fun to sort of consider as well.
Mm-hmm.
Yep. Anyway, cool paper. That was fun.
For a short paper, pretty thought-provoking, right?
Agree. That's a hallmark of work coming out of the Marie Collab.
Yeah.
Yeah.
All right. Let's see here. What do we have? We have a couple of emails. Yeah.
Yeah. Let's
hit three. Let's hit them.
Sounds good. I'll start here. So Anne writes, "Dear Professors Racaniello and Elde, many thanks for the interview with David Quammen. It was an enjoyable and fascinating way to spend my morning. I am so excited by the arrival of David Quammen's new book, The Tangled Tree. Several years ago, I took the Coursera course, The Emergence of Life, offered by the University of Illinois, where I was first introduced to the work of Carl Woese. As an aside, it was exciting to be able to interact with Nigel Goldenfeld in the course forums. So that's part of this Coursera course, is one of Carl Woese's colleagues, Nigel Goldenfeld, is helping run that. So Woese's research struck me as a fundamentally important work, and it set me on a path to read more about him and his papers. While the scientific world was aware of his work's significance, I was somewhat disheartened that the world at large was not made aware of his important contributions until his death. Yes, high school biology books adopted the 3 domains of life for students to study, but never referenced the work or the people behind it. So I am grateful that Kwamen's work sheds light on the important research and people involved. There are so many articles now written about woes. So, and Anne lifts up the March 2014 issue of RNA Biology, which was dedicated to them. She's sharing the link. We'll put this up on the website for this episode of TWiEVO. And then she also goes on to say, here's one of my favorite papers entitled by woes. The paper discusses how certain ideas can hold back advances in thinking. One of the things Nels discussed with Kwamen in your podcast. So the review paper, which is a great one from Carl Woese and I think Goldenfeld, the title of it is How the Microbial World Saved Evolution from Scylla of Molecular Biology and the Charybdis of the Modern Synthesis. So they're drawing there from Greek mythology. Do you remember this one? Scylla and Charybdis?
No. Nope. Don't.
So this is, I think it's basically, the idea is like the lesser of two evils. So anyway, so these are two mythological sort of challenges, I think, in like sea navigation or something. Anyway, so and then Anne says, share the links as you see fit. Thanks again for doing this work and giving us the opportunity to grow in knowledge about so many things. So thank you, Anne. So we will definitely post both of these. And thanks for putting that review back on my radar. I think I read that some years ago, but then kind of forgot about that. So I want to dust that off myself. I've really been thinking a lot. So, and just to say, Vincent, I really enjoyed our last podcast with David Kwamen. That was a fun conversation. It has gotten me thinking about this idea of like what holds back advances in science, or that tension between, sort of convincing peers that what you're doing is in advance, but it's not so out of the realm of like possibility, that balance. It's really interesting. Yeah. So, and thank you, Anne. That's really appreciate your support and for sharing those links.
In Greek mythology, Scylla, actually pronounced Scylla, the monster that lived on one side of a narrow channel of water opposite her counterpart, Charybdis. The two sides of the strait were within an arrow's range, so close that sailors attempting to avoid Charybdis would pass dangerously close to Scylla and vice versa. And so the idiom between Scylla and Charybdis has come to mean being forced to choose between two similarly dangerous situations. I guess it's kind of like between a rock and a hard place, right?
There you go. So here are the two evils in the Woe's metaphor is molecular biology and modern synthesis. And his notion that microbes will save us, that this will, I don't know. Well, we should all read the review.
Yep.
Yeah.
The next one's from Anthony. He has link suggestions for TWiEVO 37, which is David Kwamen episode. This Small Things Considered post might be interesting to include at the show page. Celebrating Woes, 40 Years and Entirely New Views of the Microbial World by Roberto and Elio. And perhaps listeners might find useful a list of books mentioned in the podcast with links to relevant sites. Here's one to start. The New Foundations of Evolution on the Tree of Life, Jan Sapp, and he gives a link to that. Some of these books are available online.
Very cool. Yeah, thanks, Anthony. We'll add those as well.
We'll add those, you bet. That's pretty cool.
Very cool.
The last one's yours, Nels.
Sounds good. So this is from Mike in Oregon. Hi, gentlemen. I'm curious as to the Gaia hypothesis purported by James Lovelock and Lynn Margulis. Do you think it has valid points? Also, I'm always wondering why we as living creatures have two main drives for food and reproduction, as far as I'm aware of, and I honestly can't think of any others. I know I wouldn't be here to ask this question if it wasn't somehow part of our makeup. Are we like spores on the surface of the earth, feeding the mother? A little echo there of the Gaia hypothesis.
Gaia, I think.
Gaia, yeah, that's right. From Mike in Oregon. Thanks, Mike. Yeah, well, that's a big question. And the Gaia hypothesis is a big hypothesis as well. And again, Lynn Margulis showing up in TWiEVO 37, the episode, our conversation with David Kwamen. I definitely, so actually, I think this hints again or echoes on the this very topic of like how science advances and like how radical ideas, this is a pretty radical and major idea. So in I guess the Gaia hypothesis, just to summarize that, I think it's basically, this notion that the whole Earth almost acts as a interconnected, almost like a living entity in and of itself, given all of so that sort of the biology combines with the sort of geology in a sense. So here's a quick actual definition, dictionary definition of the Gaia hypothesis: that living matter on the Earth collectively defines and regulates the material conditions necessary for the continuance of life. The planet, or rather the biosphere, is thus likened to a vast self-regulating organism. So, I guess my short answer would be, I don't know. I mean, I think it's a really interesting idea. It actually got me thinking a little bit. So we've been talking about arms races, in these evolutionary arms races, which harkens back to the Red Queen, as we talked about on TWiEVO. And in fact, the person who coined the Red Queen, this fellow Bill Hamilton. So when he near the end of his life, had this idea that actually there's a connection, sort of like maybe an example of a connection like this that would be consistent with the Gaia hypothesis. And that was that microbes cause the formation of clouds. And so not a whole lot of evidence, I would say, but the one link to that is that as people have been sampling clouds, actually, so, in the era of the microbiome, there's microbes everywhere, and there's some evidence that there are microbes in clouds. And so Bill Hamilton got this idea that would be the perfect way for microbes to spread themselves around the world, would be to hitchhike on clouds. Mm-hmm. He had some other fanciful notions. So he thought his own when he died, he thought his own atoms would hitch a ride on the clouds to the Brazilian rainforests, where he had spent some time in his career. And then he had this sort of fanciful idea that the beetles his molecules would contribute to anyway, I'm getting a little off topic there. But there are these really, I think, provocative ideas, both the Gaia hypothesis, but even just like how microbes and other, like this idea of not just symbiosis, but symbioticism. There's a great lab, again, at Vanderbilt. This is Seth Bordenstein, who's been discussing these ideas, sometimes controversial, sometimes provocative, but I think it's really worth it to step back and sort of think in these along these lines of these big ideas and see what their worth is for advancing science. So
certainly the Earth and all the life on it are big interactions, right? Certainly the life wouldn't exist without the Earth, and without the life, the Earth would be different because it's modified by living activities, right?
Absolutely.
And if you go back to the formation of Earth and the compounds that were produced early on that give rise to life and so forth, and then it's clearly interactive. Whether it's a big organism, I don't know if that's so important, but I think it just guided people to think about this in different ways.
That's right. And then the question becomes, I think that's pretty clear, that just as you're saying, that sort of all of these biotic interactions are really sort of entangled or interwoven. How much that then extends to abiotic factors, whether it's clouds or rocks or geology, all of these things, that's a deeper question.
Besides the bacteria that may be in the clouds, there are organisms on Earth that give rise to gases that encourage cloud formation, right? Am I thinking of Emiliana Huxley?
Oh, yeah.
Don't they give rise to gases that, if there's sunlight, they produce these, and when they bloom, they give off gases that then give rise to clouds, that sort of thing. I'm not. It might be Emiliana, but I don't remember. But
that sounds familiar. Yeah.
There are things like that which are clearly an example of an interaction. But whether they're abiotic interactions, I don't know. But it's interesting to think about. I think as we learn more, we find more.
That's right. Yep. 2
drives for food and reproduction. I've always thought that the main drive is reproduction. And I think humans have evolved to reproduce, and that's it afterwards. That's why when we get old, we break up. Things go wrong because you're past reproductive age, right?
That's right. Well, and then, again, it can get more complicated for humans. So is there fitness value in having grandparents around? There's some
evidence there is, yeah.
Yeah, that's right. But so in terms of drive too, yeah. So another way of framing that, this hits again on some of these themes, is Lee Van Valen, who was a contemporary of Bill Hamilton. So he, in another sort of proponent of the Red Queen hypothesis, as it was founded, he had some ideas about, the idea of energy, like just control of energy. So evolution acts over the control of energy. So food would be energy. And then with reproduction, I think, one of the bigger drivers that certainly we're focusing on is how these collisions of two genomes, so a microbial and a host collision, how that then like who reproduces the most or who has control of resources, that can all kind of mix together. So I find the interesting points being when two genomes collide, what happens, or what the outcomes from that, which involves gaining food and also obviously reproducing.
And Mike, there are people who have thought about this their entire lives, which is kind of speculating here.
Yeah, that's right.
Maybe exactly. Bill Hamilton would be somewhere to start, right?
Yeah. Well, maybe we're the spores on the surface of the earth here.
Are we spores that feed the mother? Well, if all life disappeared, the earth would still be there. It just would be different, right?
That's right.
Exactly. But, the thing is that let's say if there's no more oxygen generation, because right now the oxygen is maintained by mainly cyanobacteria and plants a little bit, right? Mm-hmm. So if all that went away, eventually all the water would go because UV light would split the water into hydrogen and oxygen, and the hydrogen would escape to space, and that would be it. There would be no more water. So right now it's kept there because of the massive production of oxygen that drives it back into water. But that's why Mars is dead, because it didn't have producers of oxygen. So if it did have water, it was all split by UV light and went off into space, and the oxygen reacted with iron, and that's why it's red.
There you go.
Why did I say that? Because there clearly is an interaction that makes the Earth what it is. There would be a red rock left if life went. It would look like Mars, I think. Okay.
Yeah.
All right, let's do some picks, Nels.
Yeah, so this is...
Before we get into some trouble here.
I agree. That's right. We might already be. But we're going to bring it back. We've got a couple of, for our picks of the week, some last-minute holiday gift-giving ideas here. I actually for my pick of the week, this is something I just did. So came across some really cool art from the illustrator artist Rachel Ignatofsky. Hopefully I pronounced that close to correctly. In her work, so she's out of LA, independent artist and illustrator. She focuses on science and nature related topics. I'll post a couple of links, one to her main website, but then also to her Etsy page where she has a shop, and some really cool, both artistic and I think informational works in prints, t-shirts, and worth checking out. So I ended up she has these little ecosystem prints. I got a set of those and gave them to some relatives last week. And there's also some really interesting ones, both art and science. So she has some women in art. She has women in science. You've got some great illustrations of Jane Goodall. You've got Rosalind Franklin. You've got Ada Lovelace. There's even a t-shirt of lab equipment. I think just a really nice artistic feel, but also information. So all these little details, like the ecosystem prints, the thing that really caught my eye was not just sort of the big picture of the ecosystem, but the down to the specific details. So just picking out a few species and even a few microbes of things that sort of hold that together. Pretty clever, I think, illustrations and neat ways of combining art and science together.
It's cool.
So check out
nice stuff.
Yeah, check out her stuff. Yeah.
Also got a bunch of t-shirts. Pretty cool.
Exactly. So you can, get on the Etsy if need be, do some expedited shipping if you have a last minute holiday gift giving emergency.
This is a good one. Chocolate is a vegetable.
Yeah.
Eat as much as you want.
There's also there's anatomy. Speaking of chocolate, there's anatomy of a good tooth and a bad tooth, comparing both of them. Also the anatomy of some plants like mushrooms and cactus. Yeah, it's a really neat.
Yeah, it's cool.
Really neat collection. How about you, Vincent? What's your pick of the week?
So I have a book by Nick Lane, who's one of my favorite science writers.
Yeah.
I think I've probably picked a few of his books before, but I'm reading an older one called Life Ascending, The 10 Great Inventions of Evolution. And what are the inventions? Well, like the origin of life, DNA, photosynthesis, the complex cell, sex, movement, sight, hot blood, consciousness, and death. So in each one, he talks about where it came from and how it evolved. And you know what I just told you about oxygen and water and all that came right from him. I didn't make it up.
No, that's right. Yep, I was just thinking the same thing.
And he's got other books. One of them is called Oxygen, where he goes into that in more detail. You know, where did oxygen come from, and there's amazing data for all these things by people who do kind of science is very different from what Nels and I do. You know, they look in rocks for minute quantities of these things that would indicate what happened a billion years ago. You know, that sort of thing. But I like it because he is a scientist and he's a working biochemist over in the UK. But he writes very nicely about how things emerged. Photosynthesis is very interesting. The complex cell, you know, how endosymbiosis led to mitochondria and chloroplasts. And so it's a lot of fun. If you like science and good science writing, you definitely check it out.
Yeah, I agree. I've had several people point out this other book he has, The Vital Question.
Vital Question is great also, yes. Energy,
Evolution, and the Origins of Complex Life. Oxygen, as you're mentioning.
Yeah, Oxygen is great. I just bought also Power, Sex, and Suicide, Mitochondria and the Meaning of Life. That was written this year.
Yep.
And I've read Oxygen, and I've read the other one you just mentioned. What was that?
Vital Question.
Vital Question. Yeah, they're both great.
Mm-hmm. Yep.
Yeah, and this one is a little older. I just wanted to see how his thinking has changed. So you can tell it's a bit older because he's talking about in terms of endosymbiosis, bacteria going into other bacteria, whereas now we're pretty sure it's an archaea that was invaded by bacteria, which led to the production of mitochondria.
Yeah, thanks in part to Carl Woese and some others, right?
Yeah, for sure.
Yeah.
So a lot of the things I say come from Nick Lane.
Yeah, it's very cool. We actually we had him. He wiggled off the hook, but he was going to come. We hosted a meeting here last spring in Deer Valley. It was on evolutionary cell biology, a regional or satellite meeting for the Society of Molecular Biology and Evolution. And Nick Lane was one of our invited speakers, and then he had to had something else come up, so he had to back out at the last minute. But yeah, it's too bad. I was really looking forward to that. But so hopefully sometime down the line, we'll cross paths with that. I agree.
Yeah,
really cool.
We'd love to get him on TWiEVO.
Yeah, we should.
That'll be fun.
Yeah, we'll do that. Put that on our 2019 roster. Hopefully we can. Yeah.
Now, there are a number of people I'd love to get. What was the guy you mentioned? Neil Shubin.
Yep. And that's a great.
We had Hopi Hoekstra, right?
Mm-hmm. That was really fun.
I would love to get Sean Carroll too, right?
Yeah, good idea. I do. Yep. Some other folks in the writing category. Some science writers that might be fun if we could do it would be Sarah Zhang and Ed Young, two folks who are doing really great work that sometimes overlaps into evolution space as well. So there's all kinds of possibilities. So stay tuned in the new year of 2019. I think we'll have some fun episodes ahead.
Stay tuned. Tell your friends. Tell them to subscribe.
Please. That's right. Yeah.
All right. TWiEVO 38, Apple Podcast, microbe.tv/TWiEVO, where all the show notes are. I know when you're driving, you can't look at show notes, but you get home and you want to see the books that we've referenced in the letters, you can go there. It's also on Google Play, but we know all of you, or most of you, listen on your phone or your tablet. You use an app. Please subscribe in the app. Really easy to do. You get every episode, and people always say, well, isn't my phone going to fill up with TWiV or TWiEVO or TWIP? No, most apps throw away ones you don't listen and only keep 2 or 3. You can modulate that yourself. And for us, it's good because we see who subscribes. We get numbers, and we can tell an advertiser, 1 million people listen to TWiEVO. Ha, I wish. But
we're heading that direction, aren't we?
We're heading that direction. We're trending upwards always. So please subscribe. And if you like what we do, please support us financially. You could give $1 a month. That would be great if every one of you did that. Microbe.tv/contribute. And send your emails, questions, comments to TWiEVO@microbe.tv. It has been my honor and pleasure to start and continue TWiEVO with Nels Elde. You can find him at cellvolution.org and on Twitter, Earlybird. Thanks, Nels.
Hey, thank you. And hats off, Vincent. What our listeners don't know is this is your third podcast of the day.
That's right.
You've already done a TWiM this week in microbiology and in immune.
That's right.
So I'm impressed with your podcasting resilience.
Yeah, don't be impressed. I love doing it, and I should be doing some other things. So I do what I love doing.
That's great. This has been fun.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. They're at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next year. Until then, be curious.